Now we are going to be studying about the skeletal muscle. So talking about the structural organization of the muscles. So the skeletal muscle has various muscle fibers and each of this muscle fiber has a protective connective tissue sheath called as the endomysium. Multiple muscle fibers together forming a muscle fasciculus. So we can see that these are the individual muscle fibers connected by having a connective tissue sheath of endomysium. Multiple muscle fibers coming together to form a muscle fasciculus. This muscle fasciculus is now having a connective tissue sheath of perimysium. And multiple fasciculi coming together to form the muscle belly, and the entire muscle belly is now going to have a connective tissue sheath of epimysium. The cell membrane of the skeletal muscle is called as sarcolemma, and the cytoplasm that is present inside the skeletal muscle cells is called as sarcoplasm. So the sarcoplasm of this muscle fiber is going to have various cellular organelles, let's say mitochondria, lysosomes, etc. And apart from this, it is going to have a large amount of myofibrils as well. So we have two different types of myofibrils. We have actin myofibrils and we have myosin myofibrils. So it is this actin and myosin which are actually responsible for bringing about muscle contraction. So each of the myofibril will have around 3000 actin filaments and 1500 myosin filaments. So it is actually this actin and myosin which are the basic units of the contractile machinery of the cell. So let's say it is going to be the interaction between these two that is going to bring about the skeletal muscle contraction. So let's see how are these myofibrils arranged in a skeletal muscle fiber. So here we can see an entire muscle belly. In the next diagram, from the muscle belly, as we can see, multiple muscle fasciculi are forming a muscle belly, and inside the muscle fiber, so multiple muscle fasciculus formed by the muscle fiber, and the muscle fiber consists of various myofibrils. Now, based on the appearance of these myofibrils under microscopy and plus based on their properties to reflect light, they have alternate arrangement. So the arrangement of these myofibrils is such that based on these properties, they give alternate dark and light bands. So it is because of this alternate dark and light band arrangement that the skeletal muscle, the myofibril as well as the skeletal muscle has a striated appearance. So we know that skeletal muscles are striated muscles. So the reasons for this striation is this alternate arrangement of dark and light bands that are seen in the myofibrils. So as I already told you, each myofibril will have both actin as well as myosin molecules. So let's see what is the arrangement of actin and myosin in the case of a sarcomere. So here what we are observing is we have an I band, right? So this I band is isotropic to polarized light, hence it is called so. Plus we can see that this I band only contains actin filaments and there is no myosin filament in the I band. Then we have the A band. So A band, it is called as A. This is going to be the darker band because it is anisotropic to polarized light, right? So A band will have both myosin filaments. So this particular filament that you are seeing, this is the myosin filament. Overlapping to the myosin filament are the presence of various actin filaments. Plus you can also see that there are some structures that are coming out. There are some projections that are present on the myosin molecule. So these projections are the heads of the myosin molecules and they are the actual cross bridges which will interact with the actin molecules, which can also be seen in the A band. In the centermost portion of the A band, we can see an H zone.